sometimes you have to be a little careful. You wind up getting introductions that you start looking around and seeing who they're talking about. But anyway, so thank you very much for that introduction. Yes, would like to spend some time this evening just talking about a few things, particularly talking about some things that are talked about and covered in the video that I made with Jim with Evidence Press. I do have my PhD in microbiology. I was a National Institute of Health postdoctoral fellow. I was also a university professor at a different university, one of those competing universities. I was a research director for a biotech company, and I'm a creationist. Now, that introduces a very interesting situation because I'm somebody that's not supposed to exist. To have this kind of scientific pedigree and be a creationist, typically this is the reaction. You get what we creationists call the face, absolute disbelief, like how in the world can such a being actually breathe and walk and talk? Because in the popular setting of the scientific community, there's not supposed to be any question, there's not supposed to be any doubt, there's not supposed to be any room for the existence of someone like me, and yet I exist. Here I am. I stand before you, flesh and blood. One blogger trying to come to grips with who I am, what is going on, they said that Dr. Ken Anderson was at one time apparently a respectable scientist. What happened? He works now for the Creation Research Society, which signifies the end of what would have been a successful and useful career. I have news for that blogger. What happened was that I understand the science, and I know the science supports a creationist view, and it's not been the end of a successful and useful career, it's only been the beginning. And I am still in it, I am still researching, I am still publishing, I am still speaking. So I did not go away, even though he may wish that I would have. Right. This is where I work. This is the research center under the auspices or under the control of the Creation Research Society. And there's our website. I think you were given a handout to introduce you to the society if you're not familiar with who they are. Perhaps you've heard of other organizations such as Answers in Genesis or Institute of Creation Research. Those are sister organizations, but we are a society of scientists and like-minded individuals. Okay? You will find a lot of the creation scientists members of our society. As a society, though, we don't have one voice, we don't have one unified idea. There's a lot of ideas within the creationist community, just like there's a lot of ideas within any kind of scientific community. The society just represents an organization where we've gotten together and said we're going to support each other as creationists. And our primary tenet is that Christ is our creator and savior, and the Bible is the Word of God, and science has the ability of showing what is true, and therefore we do not fear science. We are not anti-science, as many people will lead you to want to think creationists must be anti-science. That is a smokescreen because, in all honesty, they are scared of what we have to say. And I will show you tonight some of what they don't want you to hear. And when I say they, I mean the scientific establishment that is saying that someone like me cannot exist and the Creation Research Society cannot really represent true scientific thought. Okay. The society was founded in 1963. It's a membership society dedicated to the purpose of honoring God as creator and providing scientific arguments that counter the claims of what we would identify as the materialistic culture. Membership also supports our work and contributes to encouragement of creationists. If you recognize then, that means we are now 50 years old. We are the oldest of all of the young earth creationist organizations. So we're kind of the grandfather of all the rest of these, even though we're probably the least well known. Okay, some of our publications would include the Creation Research Society, which does actually appear in some university libraries. We also have a much more popular, less technical publication called Creation Matters. Okay, this is some pictures of the interior of our facility with electron microscopes and other microscopes and such. We're not heavily equipped. We have some equipment. We can certainly conduct some research and have conducted research and have actually published in secular literature, which again is not supposed to happen. Okay? This is often what we are posed by our culture. It's you got to choose religion or science, however you want to define religion. It's religion or science. It can be a mixture of both. You can't say I have faith in God and also that I accept scientific argument. You're not allowed to do that because supposedly they're moving in opposite directions. So what we have now is this becomes the popular view. It's evolution. However people want to define that, usually they tend to define it wrong. I define evolution as common descent. We were talking to some individuals on, I guess you'd call it farmer's market this morning, and of course they want to define evolution as natural selection, and I said that natural selection may be a process that you would claim within evolution, but you can't define evolution by natural selection. Okay? Because natural selection also works very well within a creationist model as well, which they didn't understand. That didn't make sense to them. And they said, well, how do you define evolution? I define evolution as common descent. Well, I'm not even sure they quite understood what I was meaning by that, which meant they really didn't understand evolution. When I was an undergraduate student, I took, by choice, a 4-hour college-level evolution class. My major in microbiology, I didn't have to take that. In fact, my advising professor was like, why are you taking this class? I said, because I'm a creationist and I want to learn about evolution, which kind of floored him because, oh, you're a creationist. But that was back in the 70s, so it was okay to talk about it, and we had a good dialogue back and forth. I took a 4-hour college-level evolution class. Okay. And I got an A. Now, I don't say that with the idea of showing how academically skilled I was. I tell you I got an A to demonstrate that I did understand the material. I did follow the material. I was able to provide adequate answers on the test for the professor. At the end of that 4-hour, 4-semester credit class, at the end of the semester, I sat there looking at my notes, looking at my textbook, which was the classic evolution book by Colin Patterson. And I just shook my head and says, is that it? This is it. This is what they have? This is their big bullet? This is going to blow me over? I left this evolution class far more convinced of the creationist position than I did when I entered it, which obviously was not the intent, but they made a stronger creationist out of me because I saw how shallow their arguments were. In fact, I would challenge, for my own personal life, I finished up a doctorate in microbiology. I was a stronger creationist at the end of that time when I walked down and got my diploma than I was when I entered school as a freshman. Because what I saw, what I understood, the research I did, the people I talked to, did nothing but further convince me of the correctness of the creationist position. And I am convinced that if the creationist position was allowed to the table, we would wipe through academia so fast the evolutionists would literally run in a panic. But instead, see, we're not even allowed at the table. Paul Nelson likes to do the little example where he shows the table with various pots of ideas on it, except creation. That's been pushed off. It's crumbled on the floor. You're not allowed on this table. We'll let you talk about this spiritualism and that idea and this crazy idea, but at creation, no, push that off. Well, why? Because I'm convinced they're terrified of it. Not because it's so wrong, but because it is so powerful that they know that if they have to go toe-to-toe, why do you think that creationists typically won the debates? Even by the evolutionist standards, creationists typically won the debates. Because when you go toe-to-toe and you get on an even platform, creation outshines evolution almost constantly. Okay. Here is the counter view as presented scripturally, and I would say then supported scientifically. In the beginning, God created. Not in the beginning something spontaneously formed somehow by some unknown process. Now, many people get very uncomfortable with this counter view because it doesn't fit the popular view. And so this is what we've seen too much of. Well, let's change it. So it now becomes written by man. Okay, this is maybe what Genesis says. This may be what God said. This may be what Christ said, but that's not really what they meant. And so we have a lot of human opinion enter in. I want you to think about something. In our culture, our fallen world, where it very clearly speaks about there is the existence of sin, there's the existence of fallen man that is turned in rebellion against God. If you then said, okay, in that culture, which do you think is the correct answer of where you came from, creation or evolution? What do you expect is going to be the result? Raise your hand, which is true, evolution, which is true, creation. First off, do we determine truth by raising hand, popular vote? Do we determine scientific truth? No, of course not. In fact, there were many ideas, even in science, just in the last 30, 40 years, that were very unpopular to begin with, turned out to be correct. Right? So if we had this hand-raising contest, what would we expect? Who do we expect to win? Well, of course, obviously, evolution is going to win, but remember, wide is the gate. Wide is the gate. And in this fallen world, would we expect any other answer? I get so frustrated at ministers that and other Christians that are so influenced and so intimidated by the popular vote. Well, most scientists believe in this, so it must be true. How does that make it true? Would you expect the secular world to say anything else? Would you expect them to say, oh, I don't believe in the God of the Bible, but the evidence says so I guess I'll... No, would you expect that? No, they're going to say anything but the God of the Bible, even if it means ignoring evidence. And that's what we're going to talk about some this evening, is some of the evidence. They just simply say, we can't really work with that because, see, that causes us to not be able to raise our hand quite like we'd like to when it comes to this popular vote. Okay, so I'm going to challenge these are things every Christian, really anybody, but particularly every Christian needs to think about. Okay, one of the roles of the Creation Research Society and Evidence Press is to partner with you to provide an understanding of, quote, how science supports biblical claims. Now, you've heard science says. Does science really say anything? Of course not. Science is a tool. Like a nail, like a hammer, it's a tool. I've never heard a hammer speak. I may hold it in my ear, but that hammer doesn't say anything. When you hear science says, you need to interpret it, the opinion of a scientist is. That's what that is really saying. The opinion of a scientist is. Okay? The role of science in understanding God's creation. We firmly believe in the Creation Research Society that science as a tool can be applied to look and understand God's creation and find out, lo and behold, God is there. God is speaking in this creation. Okay? To also partner with you to be able to understand misleading statements commonly made in the name of science. We're going to look at a few of those tonight. And then fourth, but certainly very important, and that is to help you understand why you don't have to check your brain at the door. This doesn't have to be something where you just got to accept it on blind faith and just push through and ignore all that you're being told that is going on and the evidence they're finding scientifically. We don't accept that at all. In fact, we accept, bring on the science. Do more. You're going to see here in a few minutes with the Human Genome Project, whereas creationists, we've been asking for more. Just bring it on. Because what happens is the more they've been studying it, the less it favors evolution. And that's what we're convinced is showing the power of the creation idea, showing that it is not afraid of science. It's not afraid of scientific exploration because it welcomes it, because it casts light on the truth of God as creator. Okay, in 2001, the sequence for all the chromosomes in the human cell was published. Now, we say all the chromosomes, we now recognize and understand that they're still having a hard time finishing up some of the sequences of some areas of these chromosomes because they're not easily sequenced. So we say they sequenced the entire chromosome, but they haven't quite yet. But in 2001, they published a sequence, said, okay, this is the human sequence in your chromosome. So we call it the human genome. Okay? A question, all right? How long would it take you to read the entire human genome if you viewed 1,000 nucleotides? Now, nucleotides, see, that's that adenine, thymine, guanine, cytosine, that ATGC that you may be seeing if you see a DNA sequence. Okay, how long would it take you to read the entire human genome if you viewed 1,000 of these nucleotides a minute? So in essence, if you read 1,000 words a minute, how long would it take you to read the human genome? The answer is that if you worked 40 hours a week, it'd take you 25 years to read half of it. That's a big book. It has so much information in it that we're only beginning to understand parts of it. Okay. Evolutionary teaching will make the claim that the more similar organisms are, the more similar their genomes would be. So for example, in this illustration, where we have an evolutionary lineage where A, B, and C all evolved from a common ancestor somewhere down here at the bottom, that we would expect that the genetic information, the genetic content, the genome of A and B would be much more similar than A and C. Because what we see is a much later branch point between A and B than we do A and C. And that makes sense from an evolutionary understanding of how lineages evolve. Right? So because of that, there's been the claim then that because human and chimpanzee diverged evolutionarily just a few million years ago, that we would still expect that human and chimpanzee would have a very similar genetic makeup. And so the claim has been that we are about 98.5% similar. So that's been the claim of you have only about a 1% difference between human and chimpanzee in your genetics, in your genome. Okay? This applies then in how people view the difference between chimpanzee and human. If you think, well, who cares? Well, here's an example where maybe you should care. This is a publication that was given to my youngest son when he was in 8th grade. And if you can read that, it says, should chimpanzees be reclassified as another species of human? Well, why are they asking that question? They're asking that question because genetically, humans and chimpanzees were assumed to be so similar. Well, we're practically just the same thing. Okay? It also carries over into politics. Here we have, a few years ago, the Spanish Parliament was contemplating giving human rights to simians. Why? Because humans share 98.4% of our genes with chimpanzees, 97.7% with gorillas, and 96.4% with orangutans. We're practically monkeys. So how do we justify saying that we somehow are different, somehow superior, we somehow should enjoy this liberty and they shouldn't? And that became the argument based upon the genetic similarity. Okay? Let's look a little bit at this genetic similarity. This is a screenshot from a website managed by the University of California at Santa Clarita. They are one of the caretakers of the human genome sequence information. And this is publicly available. Anybody can go into their website and pull this information down. Okay, what I did in this particular illustration was I pulled down human chromosome number 1. And what you have then, if you can read that, it's a little small, but what you have then is you have a comparison of human chromosome number 1 with the genome of chimpanzee, orangutan, gorilla, and I don't know, maybe 1 or 2 others up there. Okay, so this brings us then to a little excerpt of this video that Jim and I made last May, where we talk about this some.
Okay, Kevin, so I have this picture of a book from Amazon, What It Means to Be 98% Chimpanzee. So obviously this is a topic that has been going around among evolutionary circles, that there's a very close proximity between humans and chimps. And I know that you have a different view on that.
Quite a bit different view, yes.
So what's the news?
Well, I would call it the 1% myth. That really started back in a paper in 1975, before we really had much in the way of DNA sequencing. Some evolutionists, using our supposed evolutionary similarity to chimpanzee and a little bit of data and a lot of extrapolation, they proposed that we were only 1% different from chimpanzees. And so that really became the start of the 1% claim. And it started becoming popular because this fit an evolutionary concept. And as we find many times, if it fits an evolutionary concept, then it's kind of automatically taken, or shall we say it's at least taken far too easily. It's accepted far too easily. And this is one of those examples. Without much in the way of data, oh, it must be right because evolution is right, so this must be right. And it really has been, shall we say, repeated and claimed and repeated for the last several decades. And even when the human DNA was finally sequenced in the early part of the 21st century, the claim was still being made, we're only 1% different, only 1% different. But now as they're doing
There's
a
some more
much more
backtracking.
careful job of comparing and of putting the sequences together, they're realizing, okay, it's not really 1% after all. And so now there's recognition, even the evolutionist community, that, okay, maybe 4, 5, 6%, but I think that's even being way too favorable. Let me explain just one thing. You would think, okay, if we're going to compare, say the human has 23 chromosomes, and they're all in pairs, so you have 46 total chromosomes. Okay, so you'd think, well, what's the comparison of the human number 1 chromosome with the chimpanzee number 1 chromosome? But that's not how they do it. They take the human number 1 chromosome and they search all of the chimpanzee chromosomes to see if they can find little bitty sequences that line up. Doesn't have to have anything to do with it being gene structures or regulatory structures, just little bitty sequences, and see if they line up. That's how they come about with their comparisons. So
but you were also saying that there's quite a bit of difference between just the percentage of genome for the chimpanzee and the human.
Correct. There's almost 12% more DNA in the chimpanzee chromosomes than there is in the human.
And so how can you only get a 1% difference when you have 12%?
You ignore the 12%. That's the way you do it. Now, I don't know if the chimpanzee appreciates you ignoring the 12% because it probably is a little important to the chimpanzee. So that's why in 2010 there actually was a paper that finally did a comparison of all the techniques used to compare chimpanzee chromosome with human chromosome, with fish chromosome, with cow chromosome. And this paper concluded that they're so flawed in all the ways they do this that you get totally different results depending on what method you use and the assumptions of the method being used. And so the paper really suggested that we can't make a claim of what the similarity is. Now, I promise you, they're still going to be claimed 1%, maybe 2%. But we know that you can't make really any claim at all. There's just no way at the moment to adequately compare.
When I first heard this story, from you, because I really hadn't heard it from other sources, it just really made me angry because it just seems so very deceptive. I mean, it's not a little bit deceptive. It's like, this is blatant, and they're sending this message out as proof or evidence for evolution, and it becomes like an icon of evolution.
Oh, absolutely.
And it's like, well, and now they start to backtrack, and so, okay, are they going to send out an apology, do you suppose? Are they going to actually admit that they were wrong previously?
Yeah, just like they've sent out an apology for all the other things they were wrong about, yes.
I mean, this is just, I don't know if it's embarrassing for them, but it's anger-making for me.
It should be embarrassing because it shows how ready they were to jump to conclusions, how ready they were to accept false conclusions simply because they appeared to fit evolution without adequate data. So they should be embarrassed, but I don't think they will be too embarrassed, unfortunately.
Okay.
Okay, so that's a little excerpt from the video. I wanted to take a couple minutes and tell you a little bit more about that. Also maybe tease you a little bit that you want to get more from the video. Remember we were talking about that this is that website that you can have access to the genome, and it will help you do comparisons. one thing that was mentioned in the video that we want to bring out, and that is that you'll notice that, first off, what they're not doing is they're not taking chromosome number 1 from human and chromosome number 1 from chimpanzee and saying, okay, how do they match up and what kind of match do we get? Oh, we get 98.5% match, voila. That's not how it's done. Okay? How it's done is, because if they did that, this is what you'd get. And there'd be no match in there whatsoever. Okay? So to get the matching, we have to be, shall we say, more prudent. We have to be more cherry picking. Okay? So what they'll do is they'll take a certain sequence, like say this would be a portion of a sequence from human chromosome 1, and they'll tell the computer, okay, find an alignment, and the size may vary. It may be 10 bases and 10 nucleotides, it might be 15 nucleotides, 20 nucleotides, but they'll tell the computer, okay, find a region somewhere on the chimpanzee chromosomes that matches this area. Oh, okay, that one comes from chromosome 1. Ah, but to match this area, we got to go to chromosome 3 of the chimpanzee. And to match this area, we got to go to chromosome 4. See, so what we see is that you have to do this kind of selection, scanning through the various chimpanzee chromosomes. But bear in mind also, it's not like we're lining up gene to gene or functional group to functional group. Here's maybe an analogy that'll help explain it to you even better. Okay, let's take the sentence, "Methinks it is a weasel." Okay, the sentence that Dawkins used so profoundly, if you would, tried to use so profoundly in his book about the blind watchmaker, where he had that pre-programmed random generator that wasn't random and there was no way he could fail. But anyway, so I'm going to use this also, all right? Now let's take the other sentence, "We have the inks to sell." Now, how similar are those sentences? How similar is "we have the inks to sell" to "methinks it is a weasel"? All right, well, if we take the approach that is used to compare the similarity of the human chromosomes with chimpanzee chromosomes, here's how we do it. Here is INKS here, here is INKS here. Okay? Now, it doesn't make any difference that they functionally don't have the same content in the two sentences. That's irrelevant. We have the I, we have the N, we have the K, we have the S, and that's all we need. All right? Here is W here. In fact, I should have increased it. We have WE here, and here is WE here. Okay? It doesn't make any difference that one is spelling the word we, the other is spelling the word weasel. That's not important. The idea is we have WE and WE. So if we continue to do this, what we're going to find is that, lo and behold, 18 of the 19 letters match. Now, you say, but wait a minute, we have the inks to sell, we have a V and an O that aren't in the "methinks it is the weasel." Exactly, we got to ignore those. Because remember, the chimpanzee has more chromosome than we have, so you just ignore it, because that's the way the process works. So we're going to just ignore V and O because we don't need them to match up the "methinks it is a weasel." So we already have a 95% match, but we're not done yet. Because see, we can continue, we can take the I, we can bend it, we can make part of the, or we can use that to make part of the leg of the M, we can take the V out of the W and make part of the M also. So before we're done, see, we can make 3/4 of the M. So what we have in the end is we actually can have 18 3/4 of the 19 letters, and we get a 98.5% total match. That's how it's done. Okay? They're not looking for functional group to functional group. They're not looking for activity to activity. They're just taking, give me a segment and see where it matches up. So when we come back to the picture then with all the different colors, see, here's what you have. You have line up these small segments. But it's not even that tight, because in this small segment where they've lined up, you'll notice it's not a total alignment. There are 2 bases that are wrong, but that's okay, because it's close enough. So that would be considered a total alignment. Okay? We can also have this, but that's okay because we've aligned all the rest of them. So if we got extra, we just loop that out. See, so depending on the program they use, they can make all these kind of fudge factors, and it's not surprising they can be very close. See, I showed you how to take 2 sentences, nothing to do with each other, and make them 98.5% similar if I use the right paradigm, if I use the right approach. Okay? So we come back to this. If we take just a very small 1% of the human genome, and this is what it looks like as far as, notice all the different colors. Those colors are telling you that those are the different chromosomes that the program had to search through of the different chimpanzee chromosomes. So what we see is in the alignment of chimpanzee to chromosome number 1 of human, we have some from chimpanzee chromosome 1, some from chimpanzee chromosome 2, some from chimpanzee chromosome 6 and 7, etc., because they're taking bits and pieces of it. And notice also we have areas where there's no alignment at all. Even with doing this mixing and matching and this fudging, they still have areas where they can't align them. There's nothing that aligns. No matter how much they force it in, they can't make it align. Right? Also notice, and again, it may be a little hard to see on this, okay? It says chimp chain versus chimp net. What those are is two different methods of aligning the chimp chromosomes to human chromosome number 1. If you notice, they don't give you the same color patterns. Chimp chain may go to chromosome 3 and say this will fit on chromosome 1, but the chimp net program says no, I'm going to go to chromosome 6 and align that. See, so it depends on the program you're using. So it really just becomes a matter of, if I can write the program correctly, I can make it 100% match. And in fact, see, that was then brought out in a 2007 paper where they said, you know, we are having to assume evolutionary-based phylogeny, we are having to have problems with, and therefore we have to ignore what are called repeated sequences. We also are taking into account, as in, if we assume an evolutionary history, that influences how we align them. We assume human and chimp should be closer, so that's going to be built into the program. We assume human and cow should be further away, that's built into the program. So we're assuming evolution as part of our program. And also, the effects of gaps and missing data are minimized. Okay, there's a gap here, but we're just kind of minimize what that means. It's not that significant. And then, as I said, in even this paper, they acknowledge that at most there's only really about an 87.5% alignment. You don't hear that, where'd that come from? But here it is in Genomic Research, 2007. And it looks like when all's said and done, what we're really going to get is an 87.5% alignment. And even I would say even that might be being generous. Okay? Now, if some of you have heard of the organization called BioLogos, BioLogos was founded by several men who are scientists trained, who are evolutionists. Francis Collins, who was one of the leaders of the Human Genome Project, was one of the primary founders of BioLogos. And they're very adamant that I'm wrong. Okay? Dennis Venema wrote an article where he was challenging what I just said. Now, one of the ridiculous things is that what he used in his arguments is an initial 2002 chimp sequence, which has long been discarded. And that was his most recent sequence. So already it seems like he's not really up to date very well. Okay? And he goes on to make the claim, ignoring what I just told you, just pushing right through what I just told you. And he just makes the hand-sweeping gesture, by any measure, humans and chimpanzees have genomes that are highly homologous and readily interpreted as modified copies of an original ancestral genome. In which case, my response is always, by any measure, say what? He totally ignored the arguments I had just given you, as if those don't really exist, as those aren't really a problem, and just pushed right on through. Why would he do that? Well, you can only surmise. You have to ask him why he did it, but as a creationist, I would have to surmise the reason he did is because if he didn't, he's going to wind up with a conclusion he doesn't like. But then it comes back to, are we scientists following the data? Or are we imposing our opinion onto the data to make it fit what we want it to fit? So, at this time, we're going to open it up for some initial questions and answers.
I was listening to what you said about how they matched things. Mm-hmm. What if they took one of the same program and compared, say, a human being to a turtle? How close would we be? Mm-hmm.
And in fact, the 2010 paper that I briefly mentioned in the video, while I don't know that they did a turtle, they did in fact do that kind of process. What happens if we use this program? And one of the things that they were arguing was what I just said, it depends on the program. Different program will give you different results. You know, so yeah, you can compare. I think one thing they looked at is human to rabbit, for example. Okay? The proximity of the similarity of human to rabbit depends on which program you're using and what the assumptions are behind it. So it comes back again. You see, if you have the proper built-in assumptions, you can make human and chimpanzee very close. But that's the problem. What are these assumptions, and are they valid? I would chance in many instances they're not valid.
Yeah, how close did the rabbit end up being to the human?
Again, see, it depends on the program. I don't remember numbers right off. I mean, none of them are 95%, but it's going to depend on the program. See, so some of them, you know, it may be, again, I don't remember. Yeah.
Okay.
Yeah.
I don't know how to quite ask this, but when you were talking about the mix and matching between the human and the chimp, when you're matching two things that are identical, is it the normal process to do 1 to 1, 2 to 2, 3 to 3?
Now, we have to clarify a minute. Are you talking about nucleotide 1 to 1? Or are you talking about segments of nucleotides like 20, 30 bases long? Are you talking about genes that may be like 1,000 bases long? So what's the unit we're comparing?
Right, on the
Yeah.
screen, whatever
Yeah.
you were comparing.
Right. Yeah, and that
That
kind of
number one, was that-
right. That kind of comparison is depending again upon the program. What is the sequence length that is acceptable to the program to compare? And so they're not comparing nucleotide to nucleotide, they're comparing like 15 to 15 or 20 to 20.
And what should be done?
See, here becomes the problem. And even as a creationist, I say it's almost impossible to compare these because it really isn't apples and oranges. At the bare minimum, what should be done is a comparison of the functional segments. But we don't know all the functional segments because that comes back again to the junk DNA. We're finding out the entire human chromosome is functional. But what they're doing with this mixing and matching is it's not a matter of this gene compares to this gene. It's a matter of a little piece of this gene compares to a little piece of this gene, but that has no functionality related to it. So it's irrelevant, the comparison. And that's the problem they're running into. So, but it really is ultimately very difficult to compare genomes because they're just so, it's an apples and oranges comparison. And that's the problem they're running into. That's why they're having more and more struggle of, well, how do we do it? How do we approach this? Because everything they use doesn't work. Let me offer another additional explanation. In 2005, which is, as far as I know, the most recent chimpanzee alignment, when they sequenced the chimpanzee genome, it wasn't a matter of just sticking it into a sequencer and just churning out all these millions and millions and millions of bases. You have to piece it together. What they did was they pieced it together using the human genome as the scaffold. Because the assumption was, well, human and chimpanzee are so genetically similar, it ought to very easily line up. Any competent geneticist now knows that 2005 chimpanzee alignment was a disaster. Well, if evolutionary predictions were so brilliant, it shouldn't have been such a disaster. But that's exactly what creationists would have predicted, is that's going to be a disaster. Who was right? We were right. Will we get credit for being right? Of course not.
Just think about it. Think about the differences between chimpanzees and humans. Just if you were to have a picture of a chimpanzee and a human, how much similarity do we actually see? But for them to come up with this idea that there is such close proximity just isn't logical. 98.5% or 99%?
Well, you have to recognize also 98.5%, but see, there's a 5% difference between everybody sitting here. Okay, so that's where the 1% comes from. So technically, the argument was originally there's only 1% difference because the half percent is already accounted for just by diversity of human to human. So the chimpanzee is just 1% more different than that. And that's where the argument was really stemmed from, that they're practically human.
What would be the difference between like a father and a son? How would they make that comparison?
Well, again, see, that's going to be, while it'll be less than 0.5%, there's still going to be some difference, obviously. So you're going to be looking at, they're not identical. So you're still going to be looking at a fraction of a percent difference, even between father and son. Yeah, so that's why I say, just sitting here, you're going to have, approximately a 0.5% difference between you and the person sitting beside you, give or take. And that's just normal human diversity. And so that's why I say with the chimpanzee, with the 98.5% claim, see, it's really just 1% additional, now suddenly you get a chimpanzee. Isn't that amazing what we can do so much different than the chimpanzee just with 1% change in our chromosomes? Well, I think that itself is almost a miracle.
Like backwards, if I was a scientist, I would have looked the human genome versus the chimp and then done it flip-flopped. Do the chimpanzee against the human.
See, that's the problem. They're having difficulty actually establishing the chimpanzee chromosome, chimpanzee genome, because see, in 2005 they lined it up when they put it together. Because see, the way you do sequencing is you get segments at a time. Then you got to figure out, okay, this segment fits here. So this segment starts at nucleotide 1752 and goes to nucleotide 2717. And so you got to figure out how to set the segments together. Well, when you're working in the dark, you don't know where to piece these together. So that's why they use the human as a scaffold to build it on. When they realize that doesn't work, okay, back up, put the chimpanzee together on its own. That's what they're trying to do, and it keeps falling apart. So they don't, that's one of the reasons, if you notice in the video, when it was saying, when I made the comment of the 12% larger chimpanzee genome, that was based on one paper. See, it varies. What's the actual size of the chimpanzee genome? You'll never get a consistent view. Why? Because they don't yet have it lined up. In fact, you don't even get a consistent view of what the size of the human genome is, because it keeps changing some too. So how do you know it's only 1% difference when the size of the genomes keep changing? Yes.
So in the bacterial world,
yes, much simpler genome, which is
yes.
why I went there. Yeah, I wanted to study bacteria
Interestingly,
genome.
when with genome sequencing technology, it's relatively easy to get an entire genome.
Yes.
So now, when they look at one genome of a bacterium versus another one, it's assumed that if they're less than 98% similar, they are different species.
Exactly.
So,
Exactly.
yes.
Yeah.
It's beginning to contradict what they're
my
saying.
my my postdoctoral advisor would always laugh when it came to the insect world, that it was always like if the insect sneezes different, it's a different species. So speciation itself and the boundaries between different species is so arbitrary and so subjective. Exactly.
And then one other question is, if you took the human genome, and oftentimes the comparison is done to how often would you expect to find this in a random sequence. If you took the human genome, chromosome number 1, and compared it to the same number of random nucleotides that you would find in the chimp, can you find, and especially if you're looking for just little pieces, 15 base pairs, you're going to pretty often find that in a random.
You will. That's a very good point. Yeah. If you just randomly generate a sequence, and in fact, I almost think that somebody might have done that. You might be able to point me to the paper, but I'm almost thinking, a recess in my mind, somebody actually did that and said if you randomly generate a sequence and then use one of these programs, lo and behold, you still get some matching with the human genome to this randomly generated sequence.
We have time for one more question. There's a gentleman over there, Paul.
It would seem to me that there'd be no logical way to do this unless you understood minutely what the sequences meant, what they did, their function, and everything else. What percent, let's say, of the human genome do you understand step by step by step by step? Almost nothing, right?
And it is right. It is very small amount, because what we're finding, this has been one of the beauties of the Genome Project, of the consequences of it, is what we're finding is that so much of what we understood about genetics in the 70s and 80s and 90s is not wrong, it was just vastly incomplete. Let me give you one real brief analogy of the human genome. Think of it in terms of a book. Let's say it's a 2,000-page book of War and Peace. Okay, now granted, this is just an analogy, okay? If you wrote that book the way the human genome is written, you would read that book in such a manner that you would read the normal way, left to right, top to bottom, but then you would turn it over and read it again. And then you'd fold the page and read it again. And you'd fold another page and read it again. And you'd bring in another page and fold that in and read it again, so that when you were done, what you would have would be the entire work of War and Peace written on 20 pages. Not super small text, normal text, 20 pages. Now here becomes my challenge. Could you write that book? No. No human would even begin to know how to write the book. Could you even read that book? No. But you know what? That book has been written. It's called Your Genome. And yet we're told somehow that book wrote itself. See? And we're the ones accused of being anti-science, anti-knowledge, of not paying attention to the current understanding in science. We're going to move on. We won't spend as much time on some of these other segments, but another segment we talked about, or another topic we talked about in the video that I want to touch on a little bit, is the idea of what's called junk DNA. And that is the idea that in your chromosomes, in this genome we're talking about, a lot of your DNA is useless. It was DNA that's left over from when you were in your evolutionary fish stage, or when you were in your evolutionary lizard stage. So you see, the genes you had when you were a fish, the genes you had when you were a lizard, obviously you don't have them anymore, but they just didn't immediately drop out of your DNA. They're still there, but they're just kind of randomizing. They're kind of degrading. And so they're kind of the vestigial leftovers of DNA from bygone evolutionary history. So that's what the idea of junk DNA is. And of course, this is thrown out as, well, a creator wouldn't put all this useless junk in the DNA. What would be the point? Therefore, this indicates that it wasn't created, that your DNA wasn't created. It is a product of a long evolutionary history. All right? Francis Collins, which maybe some of you have heard of, he wrote a book called The Language of God, and in that he said there were long stretches of DNA in between genes that didn't seem to be doing very much. Some even referred to these as, quote, junk DNA, though he admits a certain amount of hubris was required for anyone to call any part of the genome junk, given our level of ignorance. So he admits on page 111 of this book that, a lot of this we just simply don't know about. So it seems kind of premature for us to say, well, it must be junk. And yet the irony is that 19 pages later, he's saying Darwin's theory predicts that mutations that do not affect function, namely those located in the junk DNA, will accumulate steadily over time, and this is exactly what we observe. So even though he said, we don't know a lot of what's going on, so it seems like it's kind of jumping to conclusions to call this junk, but I'm going to call it junk because it fits my calculation of what evolution would be, and therefore, see, this is exactly what we observe is we have all this junk. So he totally contradicts himself in his own writing. He then goes on to say the variation that Darwin postulated is supported by naturally occurring mutations in DNA. These are estimated to occur at a rate of about one error every 100 million base pairs. That's a nucleotide, okay, per generation. Most of these mutations occur where? In parts of the genome that are not essential, i.e. the junk. Therefore, they have little or no consequence. So he's appealing to this so-called junk that even admitted seems premature to appeal to, yet that's what he's appealing to. And then he concludes, this is exactly what Darwin's theory predict if, as some might argue, these genomes were created by an individual act of some special creation. Why would this creator put these features of junk in the DNA, is in essence what he's saying. Okay, so then in this segment of the video, Jim and I talk about this.
So Kevin, Richard Dawkins is probably one of the most prominent promoters of evolution that I'm aware of, and he was a big promoter of junk DNA. Now since the ENCODE project, there was a whole slew of articles that were published last year from the ENCODE scientists. Can you explain to us what is ENCODE and some of the background to ENCODE before we get into junk DNA?
Well, ENCODE is a consortium group of, I don't even know how many, I mean, several dozen different scientists working to take a very, very small portion of the human chromosomes, the human genome, and analyze it in great detail to see exactly what it's doing, what's being made from it, what it looks like, what genetic content it contains. And they have, over the last several years, including, as you mentioned, a series of articles just last fall in 2012 that have been talking about this small segment of DNA in the human chromosomes and just how much it's doing. Instead of it being not much activity, it's a lot of activities, a lot of function, a lot of genetic events going on. And so, to their surprise even, they've had to conclude that it's not that most of the genome is not doing something, it's most of the genome is doing something.
So what we see is that they're finding that, in fact, it's not just junk in there, that you don't have all this residual leftover stuff from your evolutionary history, that the genome's very active, the genome's very dynamic. And so, interesting point, evolutionists made the prediction based upon what they would understand of the origin of the human genome, that it's going to be filled with all this junk, all this leftover DNA from your long evolutionary history. Creationists made the prediction that's not true. You don't have that long evolutionary history. You're not going to have all this junk in there, that the genome's going to be found to be functional. And in fact, which is being found correct? The creationist prediction is being found correct. What's more, they're now also recognizing that many of these regions that were, quote, identified as junk are actually very significant in terms of health, in terms of disease, in terms of medical advances. So what we see is, in fact, that the evolutionist proposition that it's so much junk delayed medical discovery by at least 10 years, if not more. So here's a situation where we're accused of being anti-science. We're accused of slowing science down, and yet here's a clear, documentable example of where the predictions based on evolution have very significantly slowed down medical discovery. Okay? Mitochondrial Eve, mitochondrial DNA, is another topic that is often talked about in terms of showing or supposedly demonstrating genetically what's happened evolutionarily. All right, what we've been talking about so far is and we get this to work, what we've been talking about so far is the DNA in the chromosomes. That's what's considered the genome. You have all the different chromosomes in the nucleus of the cell. There's also another structure in the cell called the mitochondria. The mitochondria is where most of the ATP in your cells is made, but interestingly enough, this little structure also has a very small piece of DNA, about 16,000 nucleotides, compared to some 3 billion nucleotides of your chromosomes. So it's very small by comparison. But because it's so small, that has allowed a lot more work to be done with it for quite a few years. Like any DNA, it's subject to mutations every generation potentially. So what happened was that they started studying mitochondrial DNA and started saying, okay, can we make various estimations of how much change has occurred in the human mitochondrial DNA and what that means as far as your evolutionary history. For the most part, you pass the mitochondria down from mother to child. So we would have, for example, we could trace a family lineage by following the mother. She passes the mitochondrial DNA down. So what we would see, for example, is Here you would have the great-grandma. She would pass her mitochondrial DNA on to her children. The son, shown in the blue, no matter how many children he has, he doesn't pass his mitochondrial DNA down to his children, only the daughters do. So we can follow through the mother from generation to generation then the mitochondrial DNA. Because it is DNA, it's subject to mutation. So what happened was they used this to trace human, so-called human evolutionary lineage. And here is our fourth segment of the video of Jim and I talking about this.
I want us to talk about the concept of the mitochondrial Eve. I think that there's some misinformation out there, as I've learned again from you and your presentation, and so I want us to talk about that. My own personal experience is I have a friend who was an old Earth creationist, and he wanted me to read this particular article on mitochondria and the mitochondrial Eve and how it favors an old Earth and old time scale, this sort of thing. So I would like you to explain what mitochondrial Eve is. Of course, you know, it has to do with DNA in the mitochondria and so on. So there it is. Go ahead.
Okay. Well, the mitochondria is an organelle. It's a little structure inside of our cells, but it actually contains a little bit of DNA. Like any DNA molecule, it's subject to mutations. It's subject to having its DNA sequence altered. Like any event in DNA where you have mutations occur, we can follow those mutations, at least in a limited manner, as far as what was the original sequence versus the sequence now. With mitochondrial Eve, this arose from some work done in the late 1980s where they began to recognize that we can take the sequences of the DNA in mitochondria of people living today, and we can start comparing them. You know, someone living here, someone living in Europe, someone living in Africa, someone living in Asia, and we can begin to compare them. And in that comparison, we can begin to see, well, what exactly are the differences in all of these family groups, people that are living apart from each other that presumably haven't interbred for a while. And they came to the conclusion that, in fact, they could track back and determine that there probably was one woman whose mitochondria is what we all now have. And she became known as Mitochondrial Eve. Not out of respect of Eve of the Bible, it was a tongue-in-cheek joke. But the truth of the matter is, of course, now we can talk about Eve, you know, because it's in the scientific literature. Okay, so the idea was, we're all descended from one woman, as the Bible says.
And I go on then in the video to explain that in the 1987 study, they calculated now, we have this woman we've identified as Mitochondrial Eve. She's the source of all the mitochondria of contemporary humans. Now, they were not arguing she was the only woman alive. They just said she's the only one whose mitochondrial DNA survived to modern times. But it still fits the Bible very nicely. But what they were claiming was, but the distance between us and her, see, was 150,000 years. So they're saying she lived 150,000 years ago. Actually, it was a plus or minus. So I think it was like between 100 and 200,000 years ago. They kind of did a big plus or minus. But what was interesting was to get that age, they had to not only take the mutation rate, that is the rate that is the mitochondrial DNA is changing in human from generation to generation, but also what I would call an evolutionary time frame, also known as a fudge factor. In other words, they took and they calculated the difference between the chimpanzee mitochondrial DNA and the human mitochondrial DNA. Now you would say, what in the world, where did the chimpanzee come from? Well, remember, see, the chimpanzee and us evolved from the same common source way back when. So they felt it totally appropriate based on evolution to bring the chimpanzee genome in and compare it with the human genome and calculate the difference between the two. See, so they assumed evolution as part of their calculation to get this 150,000 years. Now, what's really interesting is when you remove that evolutionary assumption, you get rid of the chimpanzee mitochondrial DNA and just look at human mitochondrial DNA and the rate of mutation of human mitochondrial DNA, we have a problem, and that is suddenly Eve's actually only about 7,500 years old. Well, they knew that. That's why they didn't do it that way. That's far too biblical. In fact, this was mentioned in a 1998 Science article where they said, we have a problem with the mitochondrial DNA as a molecular clock. It doesn't work. The reason is because if we use it as a clock, Eve is only a mere 6,000 years old, and that's far too biblical. So this article is actually arguing to just junk mitochondrial DNA as a method because it doesn't work, because it gives us too young of age. Okay? I have a friend named Rob Carter, who is also a creationist, who published this paper in a secular journal, Nucleic Acid Research, which is one of many papers I can show showing that creationists do indeed publish. We publish in respected journals, and we publish papers in respected journals that are very friendly to creation, even though that's not supposed to happen. This paper, he published a sequence of what he determined was the sequence of, quote, mitochondrial Eve, except, of course, he was, in his mind, saying, this is Eve's mitochondrial DNA. Because, you know, each and every one of us have Eve's mitochondrial DNA. We still have it. And he was able to determine what he considered to be at least 99.99% of what was originally Eve's mitochondrial DNA. So he thinks he's getting very, very close to what her original mitochondrial DNA sequence was. In fact, to the point that he says that on average, there's about 21 to 22 mutations, as in nucleotide changes, between you and Eve. Now it varies, some people more, some people less, but on average between 21 and 22 nucleotide difference. The problem this poses for evolutionists is this is far too few to be able to spread out over such a wide time frame as Eve is supposed to have lived. But it fits beautifully with the idea that Eve only lived a few thousand years ago, as we would say the Bible says and as we would say science says. So here we have what was once held up as this demonstrates that humans are old. No, this demonstrates humans are young, just as we've been saying all along. So again, using Rob's newer data, we find that the time frame you get is probably no more than 8,000 years between Eve and us, based on genetic data. So once again, we're going to then close this portion and open back up again for questions, and maybe I'll have the answer.
So, Dr. Anderson, I would like you to take a minute and just review the three different topics that we've already talked about and just kind of summarize very quickly. These are three icons of evolution. What are we doing here with these three icons?
Yeah, that's why we chose these three topics, because they're in essence icons, genetic icons of evolution. This is very common. If you take a genetics evolution class, these would be three topics you'd probably talk about. The chromosomal similarity, the genomic similarity of chimpanzee and human as supposedly predicted by evolution because we have such a close evolutionary proximity to chimpanzee. And obviously we showed how that really becomes more a fabrication depending on the assumptions you make, the types of comparisons you do. And even the evolutionists are beginning to admit we don't have the kind of similarity we thought we had initially. And then the second icon is the idea that your DNA contains a lot of residual leftover from when you were a fish, when you were a lizard. And those segments of DNA didn't just go away, they're still there in various disrupted or degraded components, which they call junk. So the idea was that most of your DNA is residual from your long evolutionary history, but serves no particular purpose anymore. And we're showing more and more with research that's not true. It does have function, it does have purpose. It's not representing a long evolutionary history. Speaks very much then actually to that you're a recently created being. And then the third one we just finished up was Mitochondrial Eve. The idea that there's one woman who was the source of all human mitochondria, which many of the evolutionists agree with, but they tried to place a time frame, but the time frame came from assuming evolution. When you don't assume evolution, suddenly the time frame gets very biblical, and that scares them to death.
So we have another period where we're going to do a question and answer, and again, we'd like you to raise your hand so that we can pass you a microphone. I see, okay, so I guess there's a person
Yes.
back there first. So go ahead and switch it to the other microphone, please.
You showed a slide up there that was showing something was being passed down just through the female.
That was the mitochondrial DNA, correct.
Okay.
Yes.
Is that why, when you look at the geology of Christ, it's passed through the woman?
I would hesitate to make that kind of theological statement based on that. It could be. I would not personally go that far, but I couldn't necessarily argue that that wouldn't be the case. I don't know. But it is certainly that Christ would have had Mary's mitochondrial DNA. That's the way it works biologically. And if we're saying Christ was fully human, he would have inherited her mitochondrial DNA. That's the way it works.
A similar
more
question.
questions?
Why does the mitochondria only pass through the females and not?
Now there is, and I'll kind of cross my fingers here a little bit, there is some evidence that occasionally it comes from the father, but it's really confusing as to why that might and how often that happens. But for the lineage, what we just talked about, we just ignore that because that would be few and far between and it's not going to affect our overall lineage. So it just comes down to what happens is that the egg contains the mitochondria, the sperm does not. And therefore, upon fertilization, the egg mitochondria is what the embryo now has.
So, Dr. Anderson, with the evolutionary fudge factor in there, the number was about 150,000 years. That clearly doesn't explain the millions of years of evolution.
That is correct too, yes.
What is this secular argument for where all the rest of it went?
Well, of course, what they're saying is they've pinpointed back that it was at that time that the female lived who now we have mitochondrial DNA. Right. All the other females, their mitochondrial DNA died out somewhere along the line. You know, her sons killed their sons, or her daughters killed her daughters, or whatever the case is. Yeah, so that's what I'm saying is that was the break point. But obviously, they said they're not saying that was the only woman alive. They're just saying that hers is the only one whose DNA has survived to contemporary times. But I just find it so fascinating that that's exactly what the Bible says, that the Eve is the mother of all mankind, and we would all have her mitochondrial DNA. And guess what? We all have her mitochondrial DNA. It's genetically validated. We all have it. Now, if the Bible was written just by some old codger sitting in his, basement, or if it was written by a bunch of ignorant men who were just making stuff up, how would they know that? How would they have any clue to write that statement? But it's totally validated genetically because it was written by the hand of God. Yeah, wow, and that's why I love being a creationist. You get to have this kind of fun. Evolution don't give this kind of fun. We get to have this kind of fun.
Hold for 1 minute, please. Go ahead.
In the creation evolution battlefield, is it no holds barred, or I read a lot about, you guys maybe don't respect our intelligence as giving us little snippets of quotations without maybe giving us the full picture. Can you extrapolate on how those quotations can be given in context without really getting us drug into all the hairy details? Can you guys do better with that?
I think first off, in fairness, different people do different things. And so I'm not going to say that every creationist out there has always handled it most above board, made the most solid arguments, presented the best positions. Clearly some of them haven't. As far as the whole context of misquoting, I reject that because what we get called quote miners. Well, I thought that's what the scientific literature was for, was to mine and extract information out. And every single quote that I have been impressed with, like, wow, that's a powerful quote, I go back to the original paper and lo and behold, that's what the author was saying. It's not a matter of, oh, you've totally turned his words around. Now, the reason they get upset about it is because you can take their own acknowledgments, their own admissions, and make a very powerful case against evolution. Well, of course, that shouldn't be the case, so obviously you have misquoted. Okay, where did we misquote? Well, it doesn't make any difference. You've always misquoted because you shouldn't be able to do that. And so it just becomes that kind of circular bouncing around. So what it really comes down to is you can readily find in the literature world, they'll make the admission that, okay, this isn't quite what it should be. Okay, this was actually misinterpreted. This was misunderstood. But when we as creationists point that out, when we show that, oh, that's quote mining. I'll give you an example. I was speaking at a college in Canada one time, which, by the way, I guess that was a college in Canada I could go, just not in the United States. I was speaking at a university in Canada, and there was a lot of evolutionists in the crowd, and I was talking about Lucy, not my powerhouse, but she's still interesting to talk about. Okay, Lucy is a fossilized creature that supposedly represents an intermediate link in human evolution. And I was showing just the variety of artist renditions, like they give Lucy blue eyes. They give her white in the eyes. Well, see, chimpanzees don't have white in the eyes. When you look at Lucy's fossils, there's no fossilized eyes. So how do they know she had white in the eyes? Well, see, they make it up to make her more human. So I was just showing those kind of discrepancies, showing those kind of things where they just impose on the data. Well, boy, that was good. So I got accused of picture mining. I don't even know what that means. How dare you show all these pictures that contradict our thinking? What does that even mean? See, in other words, you're not supposed to be able to take and throw our own creations back at us by explaining the flaws. And so it's the same way with when I give a quote. Well, that's obviously quote mining. You're obviously taking it out of context. How do you know I'm taking it out of context? Because it supports your position, and it can't possibly really support your position. That becomes the assumption.
So you're saying that you couldn't build a little better interpretation of some of their own acknowledgments that would build on?
I think we do. I think we do all the time. The reason that the quoting becomes important is because if I as a creationist stand up and say, this is what the fossil record looks like, or this is what the genome looks like, I don't count. I'm a creationist. I'm obviously out to lunch anyway. I left my brain behind a long time ago. But if I say, this is what a respected evolutionist is willing to admit, say all of a sudden, oh, well, now maybe you got to take it a little seriously. So then, as you say, then we take and build on that and say, let's give an even more clear interpretation of the data from a creationist perspective. So we start with their admission and build upon that. And so, I'd say we do that several times.
Is it possible to build a creationist model that's not like a cartoon book of Noah's Ark that would threaten their evolutionary kind of model?
I think we really already have that. Now, in the video, as Jim was talking about, we really attacked icons of evolution. We weren't as specifically building a model for creation as we were attacking the evolutionary icons, but it gives us a starting point, because as we say, we can build as part of the creationist model using mitochondrial DNA.
because it fits very perfectly within the creationist model. See, so we have starting points, and I'd say we are building models. I say we already have very powerful models.
Do we have any other questions? Hold on, please.
So recently they said they discovered a 1.8 million year old skull. Can you explain how they come up with a number?
Well, of course, first off, let's back up a second. Regardless of the date, it's interesting. Do you know what the conclusion with the skull was? It's brought more together where they're saying it looks like what we thought were different species actually are all human after all. See, so what's happening is if you go back and look in literature 50 years ago, every single skull literally represented a different species supposedly in evolutionary lineage evolving to human. That's all gone. And they've grouped them all together now, and you don't have evolutionary lineages anymore. And that skull tightens it up even more, where there's not as much diversity as we thought there was, and so now we lose even more evolutionary history because we're all human. Now, as far as the age, see, the way that these things are dated, it's unfortunately not unlike the way they treated the genome comparisons. It's a picking and choosing. Dr. Russ Humphreys, who's a friend of mine that's a physicist, he has a talk where he puts together some 100 different, maybe even more than 100, different physical dating methods that you can use to date the age of the Earth in some manner. Of those, 90% of them give the age of the Earth as no more than 20 million years, which of course would be far too young for any kind of evolutionary history to have occurred. So only about 10% of them give the age that they want of, some 4 to 5 billion years. And his challenge is, what makes those 10% the more accurate, the more reliable methods than the other 90%? And the answer is, it gives the age you want. It's a cherry picking. Just to give a quick example, in the early 1970s, Louis Leakey, who was the son of the famous Richard Leakey, who was the son of the famous Louis and Mary Leakey paleontology pair, discovered a skull in a lake bed that his father had previously dated at about 2.2 million years of age. Well, based on the dating of that lake bed, Lewis then determined, well, the skull must be about 2.4 million years of age. But the problem was that skull was way too modern. It's known as its museum designation KMNR 1470, or just 1470. You can look it up. Okay? This skull was way too modern. So the 2.4 million year had to be wrong. They didn't challenge that maybe this modern skull is that old by their own standards. It was, there must be something wrong with the dating methods. But wait, I thought the dating methods were supposed to be objective, analytical methods that were supposed to be not subject to all this opinion and subjectivity. No, they threw them all out. All those so-called, they threw them all out, and they started over again. And you know how they wound up dating the lake bed? By pig fossils. So there's one of many examples of how did they get that 1.8, 1.9 million years? Because it's what fits their paradigm. It's not based upon this nice analytical objective measurement. In fact, I have several papers that they admit, how do we date something? Well, first we need to have an estimate of what the age is before we can date it. Yeah, where's that? So in other words, tell us what date you want and we'll get it for you. We're crashed on the floor. We got shoved off.
Well, no, but to educate the children to look at it with a lot more questions, because when I was going to school, and I'm sure maybe when a lot of these other people go to school, you only get one view. And I would never have thought to question it until I became a Christian and Probe Ministries came to my campus that I was going to school, and that just totally changed. The Bible really is true. It was really amazing. But it really angers me that the kids go to school. I saw it in my own kids, and their faith gets taken down little by little, and then they end up with no faith.
In fact, I call college, and sadly, and that's why, bless your heart again, it is such a blessing meeting you, Wisconsin. The University of Wisconsin is a great university, but oh my stars. You're sending your child to the University of Wisconsin, Satan is there. I call it Satan's meat grinder. Now, see, I've been in university settings most of my life, so I know what it's about. But Satan is there just ready to chew them up, just ready to chew them up. And it's exactly what you're referring to. They don't get this stuff. They don't hear this. Here's one thing. I challenge evolution as poor science because it leads people to make erroneous conclusions and say erroneous things. And one example of that is we constantly hear about the plight of science education in this country. It's going down, it's going down, it's going down, and what can we do? You know who gets blamed for it? We creationists. Now, isn't that amazing that the people that aren't even allowed at the table get blamed for it? It's amazing how much influence we must have when we're not even in there. Maybe the real blame lies in your teaching ridiculous science. And maybe that's where we need to start. And you start teaching good science, correct science, good way of thinking, we might see our scientific standards, our scientific understanding going up.
I was going to say, why is evolution theory? Like I said, theory is allowed to present itself. Theory, it's only a theory.
Yeah, except they won't agree with that. They'll say, no, it's beyond theory. They don't like the idea that you just tried to categorize as a theory, because as far as if you were to go to the University of Wisconsin and you were to walk into the science classroom and you were to say, I think evolution is just a theory, you'd probably get looked at like you obviously don't understand all the mountains of evidence for evolution. It's beyond a theory. It's like, in fact, I've heard professors say this, it's like gravity. It's verified over and over again. There's no question. We would love that. We would love that because we think we'd win hands down. But I think the reason we're not is because we would win hands down. How do you keep, if the Wisconsin Badgers, how's the easiest way to beat Ohio State? Don't even let them play. Just make them forfeit. We win 2-0. Yeah, go on, right? That would be the easiest way. Because if you play them, you might get beat. Oh no. So you see, that's exactly what's happened. We're not going to let you play. We just declare forfeit, we win.
There's a question back here. Going back to your point on dating, wouldn't evolutionists make the claim that creationists are doing the same thing when they look at the 10% that support their view that something is only a couple thousand years old and throw out the 90% of models that say something is millions years old?
I guess I would need an example of where we're ignoring the 90% and only looking at the 10%. I mean, if we're guilty of that, then that's something we need to look at, but I'm not aware of an example of where we're doing that. But I would certainly entertain where we have done that.
So generally speaking, then you would say that the majority of the models are supporting the older, or are supporting
I
dating.
would say from my understanding, particularly, of course, my specialty is genetics, right? What I see over and over and over again is it's not even, and I'll really go out on a limb here, it's not even 90%. It's like 99.9%. Creationists have always owned genetics. I have maintained for many years that if Gregor Mendel's work would have been as publicized as Charles Darwin's work was, you wouldn't even know who Charles Darwin is anymore. Because genetics didn't come onto the scene until after Darwinism was already fully accepted, and they had to work to synthesize it in, and it's called neo-Darwinism. It's the general synthesis. So Darwinianism was based in an absence of understanding genetics, and they had to then impose it later on. If it would have had to suffer the test of explaining genetics, I don't think it would even get off the ground. Creationists have always owned genetics. Sometimes evolutionists run up and try to plant a flag, but it falls very quickly. Creationists have always owned. Genetics has been the biggest pain for evolutionists. That's why they were so excited when the genome came out and they thought it was finally going to get rid of these creationists. And all of a sudden, it's not. The creationists are in full charge forward because the data, and that's why we keep saying, bring more data on. We are having a great time. It is just more and more and more supporting our position of exactly what creation would expect. So in my own field, I'm not aware where we're ignoring 90% of the data. I am not aware of that. I would say we are accepting it universally, and it almost all fits exactly what we would say is the case.
Dr. Anderson, would you say that the majority of the dating methods favor a creationist point of view?
Again, when we're talking about dating methods as far as the physical dating methods, if you're trying to date like the Earth, for example.
Yes, you had brought up Dr. Humphreys.
Right, that comes back to what he was showing of that 90% of them give you an age no more than 20 million years. They set an upper limit and then they go down from there.
So I'd just like to mention that I think that a lot of people are confused on this whole topic of dating. Not dating so-and-so, but dating rocks. And so, because it seems to be so profound that, well, they're always talking about radiometric dating. And that's the evidence that's always thrown out, that's brought before us. And pastors are persuaded by radiometric dating and what evolutionists say. And this is a very great concern to us. It is a huge issue. But one of the things you need to keep in mind is the world in which we live. John 3, remember John from the gospel? Verse 19, this is the judgment, that light is come into the world, and men love the darkness rather than the light, for their deeds were evil. For everyone who does evil hates the light, and does not come to the light, for fear that his deeds will be exposed. Jesus is speaking this, and he's saying something about the human condition. He's saying something about humans, that as non-Christians, they don't want to come to the light. They don't want the light to be exposed because it would expose their own darkness. And so they do all they can to spread the darkness, to hide the truth. And so they will use radiometric dating. So you ask, well, what about radiometric dating? Go ahead, he can respond to this, but really it's based on the assumption that evolution is true. And that's the thing that he keeps saying over and over and over again, and I don't know if you're getting it, is that over and over, we need to realize that the evolutionists have their evidence based upon the assumption that evolution is true.
Right. And,
as soon as you take away that assumption, they no longer have evidence.
Right. And that becomes something we want to make sure we're clear on. I'm not standing here accusing evolutionists of saying, I'm going to stand up and intentionally lie to you. I'm not saying that. And please don't ever think I'm saying that. What I am saying, though, that is in the evolutionist's mind, he's so convinced evolution is true that every piece of data is analyzed and interpreted through the light of evolution. Remember what Dubowski said, biology only makes sense in the light of evolution. If you don't interpret it within evolutionary light, it doesn't make any sense. So in other words, he's admitting it's got to fit evolution. If it doesn't seem to fit evolution, you just got to bring it in more. You got to refocus it. You got to retool it. See, so when they put an interpretation onto the, we'd say, how, like with Mitochondrial Eve. Why in the world would you bring in the chimpanzee mitochondria? Because evolution allows me to do that to get the results I want to do. And to them, it was totally reasonable because they're so convinced evolution is true. Now, the problem is, they assumed evolution to then provide an icon to support evolution. And we're saying, but when you remove the assumption of evolution, your interpretation changes. Creationists, evolutionists look at the same data. We're looking at the same human genome sequence. We go into the same, University of California, Santa Clarita website. We go into the same site, analyze the same data. Totally different conclusions. Why? Because they're focusing within an evolutionary framework and we're not. And we say, wait a minute. What about this? Oh, that can't be right. Why not? Because that doesn't support evolution. And I have heard that I don't know how many times. It can't be a right interpretation because it doesn't support evolution. I showed you very briefly the quote out of the Science Journal where they were saying you can't use the Mitochondrial Eve clock because it gives an age of a mere 6,000 years. Why can't you? Because it didn't give us the answer we want. You got to focus at evolution. Use that as your interpretation. Evolutionary interpretation tells us that can't be right.
Question over here.
No, you just addressed what I was actually going to say. It's the two different views with the same evidence that, and I guess that's what I would want students to realize. That it is the same evidence. I remember going to a lecture, and the guy was making fun of the the creationist view, and I didn't know enough at the time to, I was intimidated to say something about it, but I'm thinking you could look at it a different way, you know, the layers of the rocks and the fossils. Well, you know, when the flood, there's a worldwide flood, the water's going to stratify, the sediment's going to stratify like that, and the things are going to be buried in there, however they get buried, and sometimes the sea creatures are on the bottom, and then as animals get climbed higher and try to escape, that would make sense, you know, that's another view of the same evidence, and I just wish, that's what I want students so badly to
am
at least
I,
get that much.
Am I a totally objective, completely non-biased person? Of course not. No one is. And that also has to be understood. The scientist in the white lab coat still is a human. Okay. But one of the reasons I'm so convinced of my position is because I interact with and discuss with evolutionists all the time. And I constantly am throwing back challenges that the answer I get doesn't make any sense. And I tell them, that doesn't make any sense. And they will more or less wave their hand and say, well, you just don't understand evolution. I say, no, the problem is I do understand evolution. That's why it doesn't make any sense. And it comes back again, see, they're looking at it through the focus of evolution. Say, get rid of that. Now what do you have? And again, we'll go, since we talked about it, we'll go to Mitochondrial Eve as an example. When you remove the evolutionary presumption to it, what do you get? You get a very young Eve. That doesn't work. I think we had a question again back here.
Do you think your creationist framework could be as blinding as the assumption of evolution?
Well, in the sense that I am human, exactly. Exactly. I mean, I'm not going to stand up and claim as a human superiority of somehow my intellect over someone else's intellect. But I am going to claim that when I go toe-to-toe, when I stand toe-to-toe, when I compare arguments, when I say let's go at it, the evolutionists don't come out very well. Their arguments more or less muddle down into nothing. Their arguments, for the most part, don't add up. And I point out it doesn't add up. And often what happens is I have argued I don't know how many evolutionists to silence. Now I'm sure they would argue that, well, they just gave up because this guy's such a knucklehead. Okay? I had the I don't know if you'd call it the privilege, I had the opportunity. I don't know if any of you know who Carl Zimmer is, the self-proclaimed most intelligent man in the world. Okay. He runs a blog. What is it? Something Weave or something like that. He runs a blog. He attacked me one time. Well, I didn't take it sitting down. I challenged back. Now, to his credit, he took up the challenge, and we went back and forth at it. He made so many blunders that I point out. In this very paper that you quote showing this, it actually says this, and I quoted from the paper, and I showed from the paper. In my last post, I said, this is Carl's website. I'm going to let him have the last post. He didn't respond because he was done. There was nothing he could say. He had so blundered his position. He had so muddled it. It was a turkey shoot. I was amazed how easy it was. This is supposed to be an iconic evolutionist thinker. It was so easy. It was so easy. Just absolutely, what you're saying doesn't make any genetic sense. So those are the kind of things that convince me that I'm moving in the right direction. Again, is it because my mind's just so clouded and so fogged? Maybe so. But it still comes back to they're not defogging it. The arguments they are making don't make any sense. I showed you 3 tonight. Where was my flaw? Where did I not see something? Where did I miss something? Okay? I throw that out to evolutionists. If there was a professor of evolution right here, I'd throw it out. Where did I miss something? Now, I hate to say it, and this is going to sound like I'm picking on him, but your average professor of evolution, he doesn't know much genetics. And I don't think that's by coincidence. I think there's a reason why he doesn't know much genetics.
We have it. Hold on.
I just have 2 quick personal questions. Were you a Bible believer before you started in biology?
Yes, I grew up in
Microbiology?
a Christian home, yes. But I would say, and I do in fact talk about this a little bit in the video, I would say that when I started as an undergraduate, it was very important for me to follow the evidence. I mean, I want, in my heart, I wanted to be a scientist. That was the important thing. I wanted to follow the evidence. And I think, now again, maybe my mind's just clouded, I don't know, but I think had I been presented with what I considered evidence that just simply I couldn't get around, I couldn't ignore, that just hit me right in the face, that evolution must be true, that I would have probably gone ahead and accepted and said, I guess that's where the evidence leads, and would have had to somehow try to accommodate that. But as I told you, the opposite is what happened. I was a much more convicted creationist when I received my PhD than I was when I entered the college campus years before. Because everything I saw, everything I understood, all the discussions I had, all the papers I read, it's like, this is creation, this is creation, this is creation. And you know, that was a few years ago. It's only gotten more so since.
Second question is, have you ever had the pleasure of converting an evolutionist?
I have had the pleasure of being able to sow some of the seeds that have then come around where the person would say, it was your initial challenges that got me thinking. I wouldn't say, first off, of course, God's the one that does the converting. I wouldn't say I was there all along the way, but I have had opportunity to sow seeds. Because I used to be on a college campus, and I used to interact with college students a lot. There were several college students that come back and they say, Dr. Anderson, you got me thinking so much and challenged me so much on some of this, and I just want to thank you because I now, for the first time, like a breath of fresh air, see really what's going on here. And many of them, I will admit, many of them were Christian in their background, but they were very struggling. They were entering Satan's meat grinder. And to be able to be exposed to, you don't have to accept this. You don't have to go down that pathway. Here's another pathway. It's like, wow, I perceive, I understand. I had the pleasure, several years ago, I did a debate at University of Southern Alabama. It's always fun doing debates on college campuses, particularly because what almost always happens is very soon after the debate, the evolutionist disappears. Nobody wants to talk to him, but I get surrounded. And I had students come up and surround me, and one of them was a student in genetics. He says, you know, Dr. Anderson, I know genetically everything you said was true, but I've never heard this before. Please teach me more. And so for 30 minutes on a chalkboard, I had 20 students, and we did a creationist genetics seminar. It was wonderful. And their eyes were just wide open. It's like, we've never heard this before. We've never thought about this before. This is wonderful.
I'd like to tell a quick story similar to what you were saying. Many years ago, I was involved in a church plant, and I met a young man who was alcoholic, and he started going to AA, and he learned about a higher power. And eventually, he and I met, and I led him to Christ, to have a personal relationship with Christ. And I tell that story because I see AA as a bridge. Because if I had just simply met him, I don't know that I would have been able to lead him to Christ. Similarly, when we talk to people about the creation and evolution topic, I see it as a bridge, that it's a first step to bringing these students and young people and others who are questioning, to help them to get to that next level where now they're willing to talk about what is the gospel, and is there a God, and could it really have an effect on my life. We really want to thank you for coming tonight. Thank you so much for your attention, and it's really been a wonderful evening. Thank you, Dr. Anderson, for all that he has done, and let's give him a hand.